Let's talk about the sticker shock that comes from sample size planning. It can be overwhelming. Here, for example, you see from meta-analysis the typical effect sizes for sex differences in cognitive tasks in animal models. Based on that, what sample sizes ought to be used? They're shockingly large. And that's just to find the main effects of sex. Remember that interactions are more uncertain and therefore require larger sample sizes to detect or estimate. So what can we do? Well, one solution is right in front of us. We often need to run larger studies. It makes no sense to do affordable but bad science. And make no mistake, science with inadequate sample sizes is bad science. Another solution is to give up on some lines of research. Not everything we want to know is tractable with the resources and technology that's currently available. But there are some strategies that can really help. One example is sequential analysis. That's a principled form of run and check, where you have to spend some of your alpha on each check so that you can maintain an overall acceptable false positive rate. It has some real strengths, but it does still focus on mere detection. The really important strategy to mention, though, is optimization. Science is the art of making the invisible manifest. We magnify cellular structures. We amplify neuronal signals. We detect tiny changes in blood flow to reconstruct brain anatomy from outside the body. This is not just about instrumentation, it's about experimentation too. We strive to develop the clear and compelling tests of our research hypotheses. Sample size planning is not just bad news and a fight over resources. Sample size planning is an invitation to do the really creative and innovative work of science, to make small, intractable effects large and robust. Think of all the really fruitful and productive paradigms for mechanistic research in neuroscience: fear conditioning, long-term potentiation. These generative research programs have come from paradigms that produce large, reliable effects, and sample size planning helps point us towards developing these types of research rocks from which we can leverage the world. I can attest to this in my own experience. When I started my own lab, I began a project to measure changes in gene expression that accompany learning in aplesia. The project was funded, but we immediately began to struggle. We just weren't finding any significant differences. So we decided to go slow at first, to go back to some transcripts that had already been shown to be regulated by learning, and to adopt exactly what these other labs had done, the same protocol, and unfortunately the same sample sizes that they had used. Sometimes we'd get a significant effect, but sometimes not. And this was for genes like CREB1 that we knew definitely should be changing. It was a very frustrating year. Did we have bad hands? Did we have the wrong types of animals? Did we have bad reagents? But eventually, I began thinking more critically about the original findings and realizing that even though they were statistically significant, they were highly uncertain, and that they were providing margins of errors that were far too large for the effects we're studying. So, for example, CREB1 in the published literature had a pretty large effect size of more than a standard deviation, but the kind of standard sample sizes that were being used of just 6 per group don't provide a lot of precision for that kind of effect. In fact, in terms of power, just 60% power for that effect size. So that means even if you do everything perfectly, you should expect 40% of your experiments to quote-unquote fail. And indeed they were. Now, it was great to understand it, but we're a small lab, and we had R15 funding. We couldn't afford to suddenly collect like 4 times as many animals. So instead, we set to work optimizing. We changed the learning paradigm entirely to utilize a within-subjects approach. It was a painful decision because it meant going back to the drawing board, but it ended up being a good one because within-subject studies get you a lot more bang for your buck. We also increased the effect by increasing the amount of training we did. And then we reduced noise by speeding up our dissections. We moved the needle for CREB regulation from 1.5 standard deviations to 2.5 standard deviations. That's a much stronger signal, one that we could study within our budget. In fact, we now get really good precision from 10 animals in within-subjects design, far beyond the erratic results we used to be getting with 12 animals in a between-subjects design. I guess I'm bragging, but wow. Sample size planning is not just arcane power curves. It can help you do amazing science because it pushes you to make your research more robust and more precise, and that makes it more fruitful. So, sample size planning is an invitation to optimize your experiments, reduce your sample size needs by doing smarter, more productive science. By being very specific, because multiple comparisons hurt, by using designs that cost less in terms of the precision they can provide, by reducing noise and improving your measurements, and by doing things to increase your manipulation to increase the effect size that you can expect. So what are the key messages from this workshop? First, that sample size planning is essential. Inadequate samples are misleading and are unlikely to replicate, so you need to think carefully before and after a study about having adequate data for the effects that you're interested in. Second, that there are different ways to define an adequate sample size. I prefer the planning for precision approach because it focuses on defining an acceptable level of error or noise in my research. Power analysis is also good, but it focuses more on detectability, so it doesn't necessarily guarantee a result that will be informative for what to do next. There are other approaches out there that are worth exploring that I haven't had a chance to review in this workshop. Whatever you do, don't run and check. Don't uncritically use previous work to determine your sample size. And don't think that just because P is less than 0.05 that you're safe. Finally, I hope you'll see sample size planning as an important part of being a strategic scientist. It's about figuring out which lines of research will be the most fruitful and working to innovate to make previously intractable research topics accessible. And that's exciting. So what are your next steps? I hope that you'll go forth and sample size plan. I know that this hasn't been a comprehensive introduction, so you'll need to do some serious work learning the background and the tools, which isn't always easy. I hope the website and the workshop can get you off to a good start. If you have feedback or other resources that I should be posting, please let me know. It'd be great to crowdsource these documents to make them really helpful. Remember that if you start doing these things, you'll be in the vanguard, helping to lead our field. And on that note, I hope that you'll work to spread the good word. As a reviewer, as a thesis committee member, anytime that you're helping to evaluate science, find polite and informative ways to let your fellow researchers know that run and check isn't okay, that p-values don't justify sample sizes, that effect sizes and confidence intervals ought to be used and interpreted. Most importantly, in consuming research results, be aware of signal-to-noise issues. Compare the effect sizes obtained to the margin of error to help you determine how well the effect of interest has been defined.